Okay, we are going to change topics a little bit here because instead of talking about our gender, we are going to talk about endocrine disruptors that they also affect gender behavior, but it's not what I'm going to talk about because I was asked to talk about breast cancer. So I will start by defining what endocrine disruptors are. And then from there, I'm going to take you to new conceptions about, concepts about development and carcinogenesis, and then to the data of what is that bisphenol A is doing during fetal life that later on makes the animal predisposed to develop breast cancer. This is what I'm going to talk about, but also colleagues somewhere else in the States have found that it also applies for prostate cancer. So I will start by showing something that doesn't have much to do with the mice that I work with. Oh, well, I forgot to talk about endocrine disruptors. Sorry, I added at the last moment because I thought that maybe people don't know about them. So EPA defines endocrine disruptors as exogenous agents that interfere with the synthesis, secretion, transport, binding, action, or elimination of natural hormones. And those hormones are responsible for the maintenance of homeostasis, reproduction, development, and behavior. And the endocrine disruptor I'm working with is one that is called an estrogen mimic. It acts like estrogens by binding to the several estrogen receptors that exist. It also affects thyroid function, and it also affects androgens. But there are others that affect, for example, the synthesis or the catabolism of hormones. In any case, in order to study cancer induced during by exposure in utero, you have to move a little bit away from the normal track or the use track where people thought only mutagens will produce cancer. So you have to move to the perception of development that is older than the molecular biology revolution, and I'm talking about when developmental biology was done on the field. So in the 1880s, polyphenism was discovered, although it wasn't called like that. And I'm using this example of caterpillars that if they are fed the leaves of oaks, they look like a twig. But if they are fed, if they eat catlins, they develop this flowery pattern. So same DNA, totally different phenotype. So when we talk about the ways we look at development, the classical way from the 1950s to today was to think that the development is a genetic program, and the mother is sort of a fetal incubator and a fortress that would eliminate any influence from the external world, but in any case, what counts is the genetic makeup of the fetus. And then go to a different conception that is now being very much in force, that is the eco-devo movement, that development is an open system, that there is developmental plasticity, and that the phenotype is co-constructed not only by genes, but everything that the individual has, even its microbes, and the environment. And so it's a more of a collaborative effort, and environment co-determines the phenotype. Now, this became more accepted because in the last decade, there was much talk about humans developing diseases due to things that happen during fetal development to them, like, for example, famine, and then malnutrition increased the propensity to develop cardiac disease. So now people think a lot more about this way of looking at development. Ecological developmental biology then accepts and promotes the idea that there is no program or central control. The genes do not hold a privileged causal role. The environment plays a main role on the determination of phenotype. There is redundancy and plasticity. And organogenesis is the result of reciprocal interactions among several cell types in a morphogenetic field. So with this perception of things, we can better understand how cancer could be a problem of development going awry. And here I'm looking at the development of the kidney just to show you that you need two elements, the Wolffian duct and the nephrogenic mesenchyme, and that this reciprocal interaction gives rise to all this complexity that is not only morphological and functional, but you have a cell type diversity, so it's all even at the level of the amount of different cell types you can generate. The induction by the Wolffian duct of elements in the nephrogenic mesenchyme results in the formation of about 10 different cell types, like the mesangial cells, the Bowman capsule, etc. And the ductal system comes from the Wolffian duct. So if you put the two things in culture, you will get all these elements formed. If you put only one, it won't happen. And so with this idea, we can look at carcinogenesis when you put something like bisphenol A, for example, but we also did it with mutagens. You see that happen to be carcinogens, that is the important part. So the insult provoked by an endocrine disruptor or any kind of an oxa that will induce cancer is to disrupt the reciprocal interactions between the stroma and the epithelium, and they go in both directions, as a result of which you would have different effects in the morphology and function of the epithelium and the whole unit, the epithelium and the stroma, but we physicians still classify cancers according, if we talk about cancer, carcinoma is from epithelium, although there are transformations in the stroma as well. So continuum effect of this noxa will be to go for hyperplasia, dysplasia, metaplasia, and finally cancer. But if we look at this, then it's like development going awry. We are disturbing the relationships that makes this tissue go in one direction and not in others. So the default state of cells is proliferation and motility, like it is in yeast or any unicellular organism. And what is happening here is that by affecting the constraints imposed by the tissue, proliferation is no longer inhibited, and motility is no longer inhibited. So proliferation in the way of augmentation of the tumoral mass and motility in the sense of invasion and metastasis will occur. In any case, these two things happen in normal tissues during development and regeneration anyway, so it brings cancer to the realm of physiology and development. So what we use as a model of endocrine disruptor is bisphenol A that is found in plastics of the polycarbonate type, but also dental sealants and many different objects, including, for example, lenses, etc. And so 92% at least of people in the States have been found to have bisphenol A in urine, so that is every one of us is exposed, which makes very difficult to do epidemiological studies in humans, in spite of which there have been found very interesting correlations between exposure and obesity and other cardiovascular disease, etc. But now what we want to do is talk about the effects of this at the level of the embryo. We know that if we expose fetuses to things like DES that was used therapeutically in the 50s, cancer may arise. So an estrogen can induce cancer. In that case, it was clear cell carcinoma of the vagina, and now in 2006 it was shown that it also increased the propensity to develop breast cancer. Now, because the incidence of breast cancer has been increased during the last 50 years, much more than expected due to better detection, we have to hypothesize that that comes from somewhere and is not the genetic makeup of the population. So therefore, it's environmental, but it could be food, it could be many things. One of the hypotheses is that it's due to endocrine disruptor exposure, and particularly during fetal development. Epidemiological studies just published in the last 5 years show that gestational exposure to DDT increased the likelihood of developing breast cancer. And that happens during gestational exposure and also early after birth, but not if the exposure happens after puberty. So with these elements, we decided to explore whether or not bisphenol A, we are all exposed to it, would increase the propensity to developing mammary cancer in animal models. I should say that BPA was synthesized first. It was the first synthetic estrogen, and it wasn't used clinically, although that was the intention, because hexestrol and DS that were synthesized pretty much at the same time were far more potent. So the first series of experiments was to expose rats to environmentally relevant doses of bisphenol A. And what happened was that there was an increase on preneoplastic lesions, like intraductal hyperplasias in the animals when they reach adult age. There were carcinomas in situ as well, and they were always a low incidence, but important, of palpable cancers. So hyperplasias increase in every individual. A proportion of the animals got carcinomas that were palpable, and so this indicates that bisphenol A can act as a complete carcinogen. Now the problem is how? Exposure only occurred during organogenesis. So we decided to study that, and the first thing we did was to figure out what receptors were there. The estrogen receptors are present in the periductal stroma, and by estrogen receptors, I'm saying estrogen receptor alpha, that is a nuclear receptor, and GPR30. These two were detected there. So we did a transcriptomal analysis to see whether bisphenol A acts like an estrogen, and so it does. I mean, it looks quite more similar to ethinylestradiol than to vehicle. This is in the mesenchyme where the receptors are present. They are not present at that point in the epithelium. And interestingly, although it's not exactly the same pattern, but there is a lot in common, things become tighter when you look at the epithelium. So in other words, the pattern is more similar. And there are no receptors, so that we interpret as something that passes by is the stimulation or the effect of the stroma in the epithelium. And we decided then to study to see what are the changes that you see in the fetal mammary gland during the time of exposure. One of the things that we saw there, this is the duct, and all this is periductal stroma. So what we see here is that BPA at environmentally relevant doses removes practically all the collagen. The collagen is not organized anymore like in the control. However, it's more abundant around the epithelium, so it changes that. From the transcriptome, we look at some interesting cues, like for example, tenascin C was totally inhibited, and we see that at the protein level happens as well. So that changes totally the extracellular matrix. In addition, but I have no time to show you, there is an accelerated maturation of the stroma with increased accumulation of fat droplets. So that is adipocyte differentiation, which is very important for the growth and branching of the epithelium. So if all this was just acceleration, like we can see here, that there is more growth of the epithelium, then it would be accelerated development only. But we also found that there is a delay in the formation of the duct, the lumen of the duct, and that the organization of the epithelium was totally different. So there were changes that are not just that development was going faster. Now, what else happens here? Well, at the time of birth, bisphenol A is no longer present, so we follow these animals at 1 month, for example, and what we see is that whatever happened in utero blocks the invasion. You see here the gland is growing from here to the left, and it has passed this point, that is the lymph node, in the control, but the invasion is much more slower in the animals exposed to bisphenol A. Additionally, in the terminal and buds, we see that it's a total obliteration of the apoptotic activity that makes the duct patent at that point, and it has to do with this slowing down of growth. Additionally, we observe that the animals now express in the epithelium, many cells are progesterone receptor positive and form these clusters. Those clusters is where the branching points appear. And so after a while, at 4 months, we see that there is more branching and more formation of alveoli. So all this happens later when the bisphenol A is not there, and it's no longer there, and that is due to an exquisite sensitivity of the gland now to the effects of the hormones produced by the ovary. And that has been, we look at the dose response curve of estradiol, for example, and you need a lot less estradiol to obtain the same effect in the animals that were exposed in utero to bisphenol A. Where does it lead us? Well, we decided to look to ask the question, is this happening through the mother, or is this happening directly in the mammary gland? Because obviously during development, there are many points in which estrogens could act. One is the mother, the other is the hypothalamus, for example, the ovary, and of course the mammary gland. So we decided to test the effects of these hormones in explants in ex vivo assays, and here this was done by Lucia Speroni in my lab, and we haven't published it, but I think that is interesting to show it now. I mean, we are just submitted the paper. Here you have the control gland that wasn't exposed to BPA. This is the equivalent of E18. If the gland was exposed to 1 nanomolar BPA, that is a very low dose. So you see that the ductal tree has grown a lot. But the dose response curve is non-monotonic. A high dose, like here 1 micromolar, produces an inhibition of proliferation. And this effect is obliterated by a total complete estrogen antagonist, fulvestrant. So we can say that BPA is acting through ER alpha that is present in the mesenchyme. To our surprise, when we look at the effect of estradiol, we found that yes, the gland responds, but differently from bisphenol A. So what we see here is that there is a decrease of the growth of the duct. And this decrease is not counteracted by either, well, that jump, G15, that is the antagonist of GPR30, neither by fulvestrant. So now we do not know what mediates it, but there is an effect there, and a direct effect that we are very much eager to investigate. So, and we are investigating actually. So in summary, what we can say is that exposure to environmentally relevant doses of bisphenol A, act through two pathways. I mean, this we know that is happening, but I'm not going to explain it today. It affects the hypothalamus. For example, it affects the AVPV, that is a nucleus that has to do with the cyclicity of the female hypothalamus and the secretion of LH. And it alters several other places in the hypothalamus that will lead to a different control of the mammotropic hormones. It also acts in the ovary. But it acts directly in the mammary gland, where we see that it affects the stroma, and from there you have altered lumen formation and accelerated epithelial growth. And we know that something else is happening there. I mean, for example, the physical characteristics of the milieu are changing, so we think that it has to do with the biomechanics, this story of why you get more growth, due to all the changes in the extracellular matrix. And what we do not know and not even can infer is how all these things affect the sensitivity of the epithelial cells to estrogen and to progesterone and to prolactin during adult life. But we have shown that these three responses are altered. And we think that it's all these changes that increase the propensity to mammary cancer. We see right after puberty, there is an increase of these intraductal hyperplasias that are the places where cancer arises. So in conclusion, the fetal xenoestrogen syndrome that I only look at, talk about the cancer, challenges current reductionist paradigms and invites an integrative system outlook, bringing biophysics, cell-cell interactions, cell motility, and tissue dynamics to the forefront. So developmental genetics is not longer the only game in town to explain all the things that we observe, we and others, like altered reproductive success, autism-like behaviors, and many other different things. Perinatal exposure to BPA triggers a florid syndrome, and including diabetes and cancer. And the animals, and this is extraordinary too, at 2 days of age, their metabolomic profile is different from the controls, and this is maintained until the 6 months of age, that is the last point in which we checked. So with all this in mind, is it wise to keep exposing our offspring to these chemicals? And that is the problem. It's several decades that we know about these compounds. We knew already that they were causing a problem. 25 years since the creation of the term endocrine disruptors, we still lack a regulation here. This is happening in France. Since last year, they have a law that reduces the exposure of babies specifically, but people in general, to bisphenol A and some phthalates. So I think that here is the point where basic science intersects with policy. And one of the problems we have is that the so-called regulatory science is not science, so we have a problem there as well. Okay, I just want to mention that this is not just my work. This has been done in partnership with Carlos Sonnenschein, and there is an aspect of theoretical biology that is done mostly at the École Normale Supérieure in Paris and Université de Toulouse. And then there is our Boston group, the only, and Lucia Speroni is here, that has done all these beautiful ex vivo assays, and we have the collaborators, biophysics in Medford, live microscopy in University of Ulster, and then metabolomics in INRA Toulouse, et cetera, et cetera. Thank you very much for your attention.